and welcome you all to this third day of online workshop on molecular docking. Past 2 days we did how to select protein, how to select ligands, then how to do molecular docking. Today we are going to see how to analyze the results of molecular docked structure. Okay, this autodock. Just click on this autodock here. Then you can see the list of ligands. Then here you have macromolecule and this active site of particular protein or your uh, protein of interest. Here just click output, double click on that, you get model. Here click on molecules, just right click on this model one and save as pdb. Till this step you all did in previous session. In this session we are going to found what is the correct model to compare with our drug, because this is the standard drug one, so we have to find the correct positioned ligand, then only we have to do the comparison with drug. Okay, I'm having this PDB structure model in folder. This is this is the basic information. Then go to PyMol window. It's a Pyrex window. Now for structural result analysis, we have to go PyMol window. Yes, this is PyMol window. Here we have to open file. Just click on file, then open your saved the model file. Here you saved like this model 129, right? For yesterday's assignment you did the PDF file saved for all these models. Now I am just saying example for one model alone. Likewise you have to do for all 9. Just click on model. It says dock the structure of particular ligand which is present on the active site of protein. So for checking the correct interaction you have to click on, just first click on file, then go to open, add the protein file which you are downloaded in first day. It's an protein file. Just click on that. As per our estimation, we select this area as an active site. So the protein is docked with a particular ligand. Likewise, click on the again file, then go to open, then open the active site. Okay, here you get two different structure. Zoom. Now we have to save the specific this file, specifically this file, because this protein file containing the ligand, docked ligand and protein. After saving this file, we are going to visualize in visualization tool. Just right click on go to all, click on the all. Here remove water molecules, because that has shown some interactions in visualization tool, so you want to remove. Then add as usual hydrogens. Okay, it's all this already done. Then go to file, then save, export molecule or save molecule. Some versions we have save molecule option. Go to file, then click on export molecule or save molecule. In that dialog box you want to select only the protein, whole protein 6L ZG and the ligand molecule I am selecting. Then only we can get the ligand, docked ligand and protein. For visualization you have to save the ligand and the protein. So we want to save the full structure, we want to click all. You have to save the model as model1 result dot pdb. Don't forget to save as dot pdb, because otherwise it will not open in the visualization tool. Then in next save as type, click on save as type, just click pdb or pdb.gz. Some of the softwares may have only one pdb option, just click on that. So important thing is file name model1 result.pdb. Another step is we want to save as type pdb. Okay, now this file was saved. We want to go to Maestro. It's a role playing major role in this visualization. This is Maestro window. Through this Maestro window we can visualize the active site amino acid interaction with the particular ligand. So go to file, import structure. Now you have to import the structure which you are finally saved, the model 1 result. Please note it, model1 result.pdb file. After loading it asks OK. In between don't stop that. Okay, now you can see the whole structure. In this software you have the right click is a moving option, left click is only selection option. Okay, otherwise you can't rotate, and this using this right click you can do moving rotations. Okay, here in this site you can see the molecule. It's your ligand molecule, that is hydrochloroquine. It's this area only shown the active site ligand. It's a small molecule which are present in this particular structure. We saved the particular one, so here we got the structure. Then go to the upper side of Maestro, left upper corner you can see ligand interaction. Just click on that. Ligand interaction. Yeah, fine. You can see the dialog box. It's a dialog box open in Maestro window. This Maestro window dialog box shown. Here you can see the interaction line. But the major region amino acids 454, 53, 55 and 441 are playing major role. Here we can take it as an better model, because that previous reports are explained that arginine 454 having the important role in the spike and ACE protein interaction. This or all the amino acids are only having interaction with the arginine. Likewise, you have to do the interaction images for 9 model. Likewise, you have to do the interaction model for 9 interaction images for 9 model. So your assignment is taking the image for this image for 9 models. Some of them don't have Maestro, so they are don't no need to do that. Just take the model 1 and compare the results in Pyrex one, Pymol only. So no problem. Those who are having the Maestro, please do this assignment. Otherwise, no problem. That is not compulsory. It's optional. In Maestro, we have to do the two ligand interaction visualization for all the models. So here I am installed I'm uploaded 9 model results. So one by one I have to do for here ligand interaction. Here it's shown no interaction, so I want to take model 8. Then click on model 7. It's shown one interaction at 469. That is not relevant to us. This position having more number of interaction with the active site amino acid, like 457, 50, 56. So it's considerable. Model 6 is considerable. Then click on model 5. It's having interaction with the 471 amino acid. No interaction with the active site amino acids. So then model 4, it don't have any interaction. We won't go for that. This also don't have any interaction, so we want to eliminate. It's shown better interactions because this model 2 is having active site amino acid interaction specifically nearer to the 454. Is having less interaction. It's having shown interaction, but don't have any active site amino acid. As per our result analysis, it's don't have. Model 6 and model 2 are eligible for the interaction analysis. In this Pyrex window, you have the results for all ligands, but we want to delete other things. Just click Control and select remove selected. We need only the model 2. That interaction only having the better results with active site amino acid. So we have to take that. Then now we want to consider the best result of the drug. So this is a drug which this is the drug which we are taken as antrographlide stem learning. Okay, now we got the results for our drug antrographlide, right? Okay, where we have higher value, very less value, that is shown a better result because we got minus 4.3 for hydrochloroquine Now we got particularly for andrographite we got minus 5.5 minus 5.1. The less energy utilization is shown the better interaction. So it's a better as per the scientist report, the andrographite is the best compound for to treat corona. So this in silico study also explained that. Now what is the output we want to take means here we have go to Autodoc, just click on active site, the output of particular andrographite output, your ligand your whatever ligand you take, just double click on that output. Okay, go to molecule. Here we have the model 9 models. Now we want to delete everything except the our 2D to model 2. Remove from screen. Just active site remove from screen. This is also just unselect the everything for result comparison. Zoom at this. Just one by one we have to select. This is not fit in the particular place, so it's not eligible. The model 1 of andrographite is not eligible. Model 2 is partially eligible. Model 3 is not eligible. Model 4 is not exactly in that place. Won't take. 5th one, 6th one is not in that place. 7th one not in place. 8th one is not also in place. So first 1 and 2. First it's having the better interaction. It's also it's present in the same place of that hydrochloroquine. So we have to take that. In this model also we have to take the model 2. Just right click on that result, right click on the model 2, save as PDB, then save as drug result .pdb. Don't forget to save as a .pdb. Okay, this is the final output of the Pyrex. Just comparison of which is the exact position of that ligand bound ligand in the active site amino acid. So this first one is for result of the hydrochloroquine. In that same place we got the interaction of the andrographite. Just we have to take this for basic understanding. Then go to PyMol window. In this PyMol window, first we have to set the display background and white. It's a very important for result interpretation. Then go to file, open, just open the first open the protein, whole protein. Then open the active site of particular protein. Then open the results which now we are saved, right? Drug result, and open the model 2 result. Now in this page we got the whole protein, active site, and the ligand molecules that which are the model 1 2 result is a standard result. The 2 drug result is our drug selected ligand result. Now for this in this we have to first remove all the water. Then we have to do color changes. Okay, first the protein color should be in wheat. For paper publication only I am doing this. Active site should be in grayscale. Active site should be in surface. This surface like structure only shown the better output for our result interpretation. Here you can see the ligands which are bound at the active site inside of this. This is the result for the standard and drug comparison. Okay, in the same site, the drug and our standard are bound, so we can explain it is an active site of it's a better interaction. We can take it this image for our interpretation. Here now we have to find how to interpret the drug and ligand. Just go to table, then docking result. Here I did the docking for, here you can see minus this is the result for our andrographite, this is result for our drug. So sorry, this is result for our standard drug, this is our andrographite result. Comparatively this andrographite shown better result, it's an our output. Sometimes some other compounds may be below this value, so that is best lead in this active this screening. So for visualization, first you have to download this model first standard, then visualize the model and the protein complex formation in PyMol. Then go to Maestro and see the interaction. After the interaction finding, best interaction shown ligand model is taken as a standard. So now we are taken the model 2 is a better model. So this interaction site, based on in this interaction site, we have compare the our drug molecule. Then after just double click on this output, we got this 8 values. In this 8 values, unselect the others. One by one, we have to select the one. We have to see the position of the particular lead molecule. Which one is shown the best match the with the already known drug means that is the best output. Then just right click on that output, save as PDB. After that, after this step, we have to go to PyMol. In PyMol, we just visualize what are the visualize in the better form for paper publication and some interpretation. We have to do the PyMol visualization. For result, we have to save the this table result, then Maestro result and PyMol result. Thank you everyone. This is the whole information about molecular docking. This is the final step. If for after confirming the drug and or ligand drug standard drug and ligand, we have to do molecular dynamics simulation. That is the another major part of molecular bioinformatic computer based computational drug designing. In this computational drug designing, its molecular docking is a basic one. Here we can screen the best lead from n number of pooled number of samples. So it's a best method. Using this molecular docking method, we have in this current scenario, we can screen the drug for corona viral infection. This is in this video we just explain about the spike protein active site and protein targeting. Likewise, you have to do for if you are like do some other work now, please take the main protease, that is M.Pro, and another one is RNA dependent RNA polymerase. This two was another drug targets. One should target multiple protein, so that is the better result. Nowadays, many drugs are designed based on the multi-targeting drug, because if the drug is targeting one particular site, means that is not effective. So we have to design the drug with the multi-target. If for this corona drug designing, please do the same viral protein inhibition for other proteins also. This after this uh, different protein target molecular docking work, you can get three different results, three different number of lead compounds. Which one is come in the um, comfort for all three, means you just take that particular ligand, because that particular ligand only can target all these three target. If you are running the molecular docking for all three proteins, means in for different protein, means the lead molecule should be shown higher inhibition on whole three. If that whole three proteins can be inhibited by the lead, means that is multi-targeted drug. Thank you everyone. For any clarification, please comment.